Welcome back to Introduction to Chemical Engineering. What I'm going to talk about for the next two videos is the second of our biological product, and this second product is insulin. And some of you might have heard about what insulin is. So again, we're going from the product. This time the product is insulin. We're going to look at some of the biological aspects of it, in this particular case, the disease, if you will, which helps, where insulin is actually used to treat it, and of course the engineering of the production of this insulin. Okay, so what insulin is a protein, a peptide as it were, a small protein, and it helps to transport glucose from the bloodstream into the cells. Look, we all need energy, and glucose is one of the important sources of energy. And if the cells don't get energy, don't get the glucose, they cannot make the energy in order to survive. So, and insulin transports the glucose into the cells. And so, a little bit of history. Years ago, people discovered that sugar itself was deadly to some patients. If they ate sugar or maybe too much sugar, they essentially it was toxic to them almost. They actually died. In part because of starvation, because the sugar could not get into their cells, and in part because the sugar in the bloodstream essentially broke down into other nasty things that essentially killed them over time. So diabetes is an example where sugar is in fact deadly, unless there is a way to help transport them into the cells. But as I said, if the diabetic patients do not have enough sugar, they will essentially starve, and that's not a good thing either. So what they discovered years ago was that there is this organ system called pancreas, and it apparently in autopsies of diabetic patients, it looked different. And so the theory was maybe the pancreas has something to do with diabetes, and it turned out to be right. And then Paul Langerhans in the late 19th century discovered the pancreatic tissue function, and he called that, in fact, that's what they call the isolates of Langerhans, it's called. And then a few decades later, Minkowski and Mering, what they worked on those known hypotheses and says, what if you were to ask the question, is the pancreas directly involved in diabetes in some way? That is it making something to allow for the transfer of sugar from the bloodstream into the cells? And the answer was yes, they actually discovered that. So the pancreas is creating something, digestive juices, something to help with the diabetes. Remember, they didn't know what it was, but they knew that it was making something, and that is important. And then that in turn led to experiments by a group in Toronto, a physician, I think it was a graduate student, and their hypothesis was, as the others' hypothesis was, that pancreas makes something to help the diabetics. And so they isolated the pancreatic cells and injected some of the cells into diabetic dogs, and dogs survived. So here's a clear case in which a hypothesis led to an experiment confirming the hypothesis. So they knew that the pancreas was making something that is important for diabetics. Then the question is, okay, so what was in those pancreatic isolate cells that was helping save diabetics? Was it the cells somehow, proteins to the cells? What was it? So it took a while before people figured out that it was a hormone, in this case insulin, that is secreted by the cells that's responsible. And so the first attempt at trying to make this quote-unquote drug was to essentially take pancreatic cells and grow them, and essentially then recover the insulin from this culture, as it were, from cattle, and then continue purification. In fact, the initial insulin that humans actually used for many, many years was actually from isolated pancreatic tissue from cattle and packaged as drugs for humans. The first human trial for diabetes was in 1922. He received insulin, a 14-year-old boy, regained appetite and survived. And then there was a Nobel Prize of Medicine. I think one of these gentlemen was recognized, I forget which one is which. You might want to look up who got the Nobel Prize for medicine in 1923. So discovery of insulin. So then the question is, well, making insulin from isolated Langerhans is a good strategy, but what if we need to make more of it? Do we just harvest more pancreatic tissue from cattle and stuff? Is there another way? Is there another easier way to do it? And of course, it's not just sufficient to produce them. You've got to collect them and purify them. And Eli Lilly, as it says, started doing this years ago. And one thing that the physician discovered very early, while insulin was definitely a wonderful drug, because it was from an animal source, it was causing some immune response in humans. And that caused some problems. And the question became, is there a way around that? That is, how would you make insulin without the nasty properties of the animal source of the insulin? And that, of course, was made possible by the genetic engineering revolution that happened in the early part of the 20th century, where we understood about genes and the engineering of bacteria to make genes and so forth. And there's a gentleman here. I'll leave this. I'll come back to his picture a little later. His name is Fred Sanger. He was the first to determine the sequence of the insulin protein, insulin peptide. And he got a Nobel Prize for that. And he got a Nobel Prize for another discovery years later on sequencing the genes and so forth. So the genetic revolution is now making it possible to make insulin that does not have the nasty properties of insulin from cattle, pancreas, and so forth. It's fascinating. If you are interested in looking at trying to understand how insulin works, I provided a link, YouTube video on how insulin works. It's a pretty nice video, actually. It tells you the difference between type 1 and type 2 diabetics, the difference between insulin, not having enough insulin was the right type of insulin and so forth. And then a few facts about the insulin that's actually mentioned at the YouTubes they can actually watch.